Hello everyone, and welcome to Lecture 2 of Unit 3. In this lecture, we will be talking about one of the ways that we can control microbes inside the human body. As usual, you have your lesson objectives. Please let me know if you have any questions on them, although again, hopefully by now you know how to use them to your advantage and know how they work for you. So in our last lecture, we talked about the ways that we control microbes outside the body to help prevent infection or disease and prevent decay of things like food. In this lecture, we will be talking about the chemicals that we use inside the human body that are geared towards preventing infection or containing infection once it has developed. Those chemicals are broadly called antimicrobials. So an antimicrobial is some kind of chemical that kills a microbe. Within that very broad category are subcategories called antibiotics, antifungals, antiprotozoals, antihelminthics, and antivirals. Now, typically by their name, you can pretty much figure out what they're going to affect. Antibiotics technically kill bacteria. Antifungals go after fungi. Antiprotozoals kill protozoa. Helminthics or antihelminthics kill worms. And then antivirals stop some kind of process of viral replication. In this lecture, we are going to focus mostly on the antibiotics because that is the biggest category of antimicrobial medication that is prescribed and that is given to patients, but we will talk a little bit about some of these other types of antimicrobials as well. Antibiotics are the first group of chemicals that we will talk about, and technically, an antibiotic is a chemical that kills bacteria. Now, there is some gray area here, there is a little bit of wiggle room, and sometimes a chemical actually has more than one type of microbe it can work against. So there are some antimicrobials that are technically antibiotics as well as antiprotozoals, meaning they will kill bacteria and they'll also kill certain protozoa. But technically, kind of from a technical definition standpoint, when you hear the term antibiotic, you should be thinking about a chemical that is geared towards killing a bacteria. So when you hear someone has been prescribed antibiotics or they're going to be prescribed antibiotics, typically, at least 99% of the time, that is because they have a bacterial infection that their healthcare provider is trying to help them get rid of. So antibiotics actually have a long history in humanity. Primitive forms, and I do put that in quotes because there's some gray area here. But these early forms of antibiotics were actually discovered thousands of years ago in things like certain plant extracts, in moldy bread. We have evidence of ancient cultures having figured out that they were able to treat or sometimes prevent certain diseases by using certain plant extracts or using the mold from bread or other types of food products that had started to decay. Our current forms of antibiotics, so our contemporary forms of antibiotics, are ones that started to be developed in the early to mid 1900s. Now, the history of our current day antibiotics goes back farther than that. In the 1800s, when people started to really understand germ theory and understand that these microbes could cause us harm, lots of scientists and physicians were working on developing chemicals that would kill microbes or at least stop them from growing. But it took some time, and so kind of our current iteration of antibiotics really got going in the early to mid 1900s. And originally, all of our antibiotics came from other living organisms, specifically 2 genera of bacteria and 2 genera of fungi. Around this time, it was discovered that certain types of bacteria that came from the groups Streptomyces and Bacillus, and certain types of fungi that came from the groups Penicillium and Cephalosporium, could produce chemicals that would inhibit the growth or kill other types of microbes. So these types of organisms typically grow in the soils, and it can get pretty crowded in the soil. And so for them, they would produce these chemicals that would kill or inhibit the growth of other nearby bacteria. This would give these bacteria and these fungi more space to live in, access to more nutrients, and just basically ensure their survival. So these types of organisms, the organisms that were the Streptomyces, the Bacillus, the Penicillium, and the Cephalosporium, they just produce these chemicals naturally out in the soil, out in their natural environment. And those chemicals killed nearby bacteria. And through a lot of work, various scientists were able to harvest these chemicals. They were able to take these types of bacteria and fungi and grow them in the lab and force them to produce these chemicals. And then they were able to extract those chemicals and purify them and put them into some kind of form that would work in the human body, whether that is in a pill or in an injection or in an IV solution. Whatever it was, they were able to extract the chemicals that these microbes would produce and put them into some kind of medication so that people who were suffering from bacterial infections had some help while they were trying to fight those infections. Now, these days, we have a lot of other antibiotics that have been developed and are in use. Some of those antibiotics are from our original classifications or classes of antibiotics. Some of them are closely related to those antibiotics, although not the absolute originals. And some of them are entirely new. Either they came from another type of bacteria or archaea or fungus that has been discovered, or they've been completely synthesized in a lab, or there's some kind of combination of a natural antibiotic and some kind of synthesized chemical. Either way, whatever type of antibiotic we're using, these kinds of medications tend to work in a few certain ways to kill bacteria. And over the next several slides, we will go over how these medications actually inhibit the growth of bacteria or just straight out kill them. Before we get into the exact details about how antibiotics kill bacteria or stop their growth, we are going to talk a little bit about what we call antibiotic spectrum. Some antibiotics are very good at killing a wide range of bacteria. And they're good at that because they work on structures that basically all bacteria have. So some antibiotics are very good at doing things like inhibiting ribosomal function or interfering with DNA or RNA, or in some way just interfering with some kind of trait or characteristic that almost all bacteria have. These are called broad spectrum antibiotics, and they are typically effective against all kinds of bacteria. They might be effective against gram-positive cocci, gram-negative cocci, gram-positive rods, gram-negative rods, the spirillum and the spirochetes. Basically, these are antibiotics that have a very good chance of being able to kill or stop the growth of a wide variety of bacteria because they do something to interfere with a structure that is common to almost all bacteria. Narrow spectrum antibiotics are the opposite of that. These are antibiotics that are targeted to work on just one or a few types of bacteria because they target a very specific structure. So an example might be that we can develop an antibiotic that really only works on gram-negative rods because of the way their outer membrane is structured. Or we might be able to develop an antibiotic that only works on gram-positive cocci because of their cell wall, because of that peptidoglycan that they have in there. Or, kind of any trait that a bacteria has, we could develop an antibiotic that specifically targets that trait. Those types of antibiotics will only work on those types of bacteria that have whatever trait it is that we targeted. Now, there are benefits and drawbacks to each of these types of antibiotics. Broad spectrum antibiotics are great in situations where someone has an infection. There may not be a lot of time to help them out. So maybe they're very sick, or the healthcare provider needs to prevent them from getting very sick. And so in one of those cases, if there's not enough time to maybe do a culture or figure out exactly what it is that person's infected with, they can be treated with a broad spectrum antibiotic. And hopefully, ideally, that antibiotic will at least do enough in the meantime to help them survive this maybe dangerous period. On the other hand, broad spectrum antibiotics will also do a lot of damage to the microbes that are called normal flora in the human body. So we've talked about this a little bit before, but basically the human body is covered with microbes. It's covered all over the surface. It's covered inside as well, so in the GI tract, in various locations throughout our body and on top of our body, we have bacteria that are just creating this protective film around us. And those bacteria are very, very important in terms of making sure that we are not harmed by bad bacteria. So unfortunately, broad spectrum antibiotics will cause damage to the normal flora or the normal bacteria that we have on our bodies. On the other hand, a narrow spectrum antibiotic, like we've talked about, will only kill one or a few types of bacteria. So if we are able to know exactly what a person is infected with, and we have a specific antibiotic that will treat that, we can use that so that we target that specific pathogen or that specific bacterium and hopefully do a little less damage to the normal flora in our body. The drawback to that, or the difficulty with using these types of antibiotics though, are of course we have to know exactly what the patient is infected with. And sometimes that's either very difficult to do, or like I said, there's just not enough time to figure that out because they're very sick or they're about to get very sick and they just need something to help get them through the very dangerous period. So antibiotics tend to fall into one of these two categories, either broad spectrum or narrow spectrum. There are benefits and drawbacks to both. And which type is used really just depends on the situation, whether we know exactly what the patient is infected with, how sick they are currently at the time they're seeking treatment, and the availability of specific antibiotics to treat specific types of bacterial infections. So now that we've talked a little bit about what antibiotics are, the history of antibiotics, and the types of spectrums of antibiotics that we have, we are going to go ahead and talk about antibiotic function, meaning that we are going to start discussing the specifics about how these chemicals that we call antibiotics actually start to interfere with the bacteria that could be infecting us. We can group antibiotics into 5 main categories. We have antibiotics that work on the cell wall, antibiotics that work on cell membranes, antibiotics that work on DNA or RNA stability for the most part, antibiotics that inhibit protein synthesis, and antibiotics that inhibit some type of metabolic pathway. We are going to talk about each of these on their own separate slide, but just for now, what I want you to look at on this slide is that cell wall inhibitors are divided into 2 subcategories: the beta-lactams, which are composed of the penicillins and the cephalosporins, and just various individual antibiotics. Cell membrane inhibitors are called, for the most part, polymyxins. DNA and RNA inhibitors are the fluoroquinolones, mostly. The protein synthesis inhibitors are the tetracyclines, macrolides, chloramphenicol, and aminoglycosides. And then the metabolic pathway inhibitors are sulfa drugs. Our first group of antibiotics are the cell wall inhibitors. Cell wall inhibitors are antibiotics that damage links in peptidoglycan or prevent the links in peptidoglycan from forming. So as you remember from Unit 1, peptidoglycan makes up the cell wall, and the strands are linked together. So these in the purple are kind of the backbones of peptidoglycan, and these chemicals on the inside, they are sugar protein chemicals, they hold the strands of peptidoglycan together. The cell wall inhibitors typically interfere with these middle strands. So cell wall inhibitors will do something to interfere with what we call the cross-linking of these sugar protein molecules. And when we interfere with that, it means the backbones of peptidoglycan, the ones right here in the purple, can't stick together, and therefore the cell wall can't stick together. Now, these types of antibiotics typically don't kill bacteria, they just stop their growth. So these are what are called bacteriostatic antibiotics. Because instead of killing the bacteria, which would be a bactericidal antibiotic, these are ones that just stop their growth and basically prevent them from making any new cells, and therefore the bacterial infection can't continue to grow in the human's body, and the human or the animal in certain cases can take care of the infection on their own. So these just stop the infection from getting any worse and rely on the immune system to do the rest of the work. Now, there are two subcategories within our cell wall inhibitors. There are the beta-lactams, which is its own class or subclass of antibiotics, and they account still these days for about half of the antibiotics that are prescribed. So about 50% of the time an antibiotic is prescribed in the United States, it is some kind of cell wall inhibitor, and it is either a penicillin or a cephalosporin. These are called beta-lactams because of this special ring that is found in the antibiotic. So I'm highlighting it in blue right now, or kind of tracing it in blue. This ring-shaped molecule is called the beta-lactam ring. And this is a kind of the functional part, or a functional part of the molecule that is really useful in helping prevent the peptidoglycan strands from being able to stick together. So it's this beta-lactam ring that gives the function to the penicillins and the cephalosporins. Now, there are also some other cell wall inhibitors that do not fall into what we call the beta-lactams. They're just kind of individual antibiotics that don't fall into this class, but still work to inhibit the cell wall formation in bacteria. And these are vancomycin and bacitracin. Both of these, even though they're lacking this beta-lactam ring, they are very large molecules, and they still do a good job of interfering with the cross-linking between the peptidoglycan strands. So they're very good at inhibiting the cell walls. Most of the time, cell wall inhibitors work best on gram-positive bacteria because they have a lot of peptidoglycan. However, these days, with some very specific cell wall inhibitors or just some modifications to our original ones, we are able to make them more effective against gram-negative as well. But most of the time, cell wall inhibitors are used with gram-positive infections. Not always, but most of the time. Our next group of antibiotics are the cell membrane inhibitors. In humans, the cell membrane inhibitors that we use typically fall into the class or the group that we call polymyxins, specifically polymyxin B sulfate. This type of antibiotic works best on gram-negative cells because of the way it attaches to the outer membrane and pulls it apart. These antibiotics have what we call detergent activity, and remember we talked about soaps and detergents in our last lecture about how they work. And really what they do is they attach to this outer lipid membrane on the gram-negative organism, and they grab onto it and start to pull it apart. What this ends up doing is damaging the whole structure of the cell. Usually these types of antibiotics are bactericidal, meaning they kill the bacterium, and they can be very effective against gram-negative infections. However, the drawback to them is they are fairly toxic to humans, especially in kidneys. So these types of antibiotics can do some damage to the kidneys if they are given internally. And sometimes they have to be given internally and either through some kind of IV solution or some kind of injection. But if it's possible to avoid using them internally, it's better. However, they are typically found in over-the-counter antibiotic ointments. If you pick up what's called triple antibiotic ointment, the brand name that you usually will see is Neosporin, but there are other non-brand names. And these ointments have 3 different types of antibiotics in them. One of them is usually polymyxin B sulfate. They are great at dealing with these gram-negative bacteria that could be on the skin and helping get rid of those infections. And if you just use them topically, they are not nearly as damaging to our kidneys because they don't get very far into the body. Our next group of antibiotics are the DNA and RNA inhibitors. Primarily, these work on destabilizing DNA, although they can have some anti-RNA activity as well. For the most part, they work on DNA, but occasionally they can start to interfere with RNA too. These antibiotics are the quinolones, the fluoroquinolones, and the rifamycins. I've got some examples here. So ciprofloxacin, or just Cipro is kind of how it's commonly abbreviated, rifampin, and nalidixic acid. These are some of the most common DNA RNA inhibitors out there. And what these types of antibiotics do is they typically bind the enzymes that are involved in DNA stability, especially stability involved in helping go through replication, so binary fission. Most of the time in gram-negatives, these antibiotics target DNA gyrase. In gram-positives, they target topoisomerase, although there is some cross-activity occasionally. These antibiotics can either be bacteriostatic or bactericidal, depending on which specific type of antibiotic we're talking about. So within this broad class of antibiotics called DNA and RNA inhibitors, they can either work to kill the bacteria outright, or they can just stop its growth. These are typically fairly broad spectrum antibiotics because all bacteria have DNA and RNA. So most of the time they work equally as well against gram-positives and gram-negatives and are one of our groups of antibiotics that can be used in a lot of different situations because they work that way. Our 4th class are the protein synthesis inhibitors. And there are actually a lot of antibiotics that fall into this category. Protein synthesis inhibitors do one of 2 things. They either interfere with the mRNA being read at the ribosome so that tRNAs cannot be brought over and matched and amino acids can't be strung together, or they actually interfere directly with the peptide bonds between amino acids. The way they work just depends on which specific antibiotic in this group we're talking about. And this is another one where the antibiotic could be bactericidal or bacteriostatic, just depending on which specific type of antibiotic we're talking about. There are some very common protein synthesis inhibitors, and those are the aminoglycosides, the tetracyclines, chloramphenicol, and the macrolides. And for each of these groups, except chloramphenicol, there are multiple individual drugs that fall into these categories. So one group of protein synthesis inhibitors are the aminoglycosides. These are oftentimes reserved for very serious infections because they can be a bit damaging to the human body. Some common examples of aminoglycosides are streptomycin and gentamicin. So oftentimes these are given in situations where another antibiotic might not work, or we have a person with an infection that is deemed serious enough that it's time to call in, the big guns and use one of these bigger antibiotics. The tetracyclines are another group of protein synthesis inhibitors. The benefits to these are that they are cheap and easily available. The drawbacks are that they can cause tooth discoloration in young children, and bone problems in fetuses, and they may cause GI problems in everyone. Basically, if tetracycline is given to someone who is still developing their adult teeth or they're giving to a developing fetus, it can interfere with laying down some of the important parts of the tooth or interfere with certain parts of the bone. So they tend to be reserved for situations where the child is over 8 years old or a woman is not pregnant. Examples of common tetracyclines are a drug called tetracycline and one called doxycycline. Chloramphenicol is another protein synthesis inhibitor. It is just one drug. It's not like a group of drugs like these other ones are. This is oftentimes a drug of last resort. It is, or at least it can be, a very damaging drug. There are a certain number of people who, if they are given this drug, they will develop something called aplastic anemia. Basically, they just stop making red blood cells. We never are sure who that's going to happen to. So there's no way, at least at this point in time, to predict who might develop aplastic anemia after having been given chloramphenicol. So oftentimes this is another one that's reserved for very, very serious situations. And sometimes it's reserved for a situation where there's nothing left to treat an infection. And if the person is not treated and cured, they're going to die of the infection anyway. So something like a brain abscess or something really, really serious that they say, well, let's go ahead and risk giving them chloramphenicol because if they don't get treatment, they're definitely going to die. And hopefully they won't develop anemia later. The last group of protein synthesis inhibitors are the macrolides. The good news about the macrolides is they're fairly gentle antibiotics. They can be given to a lot of different types of people, whether we're talking about potentially young children or babies, sometimes pregnant women. They can be given most of the time to anyone without a lot of serious side effects. They're oftentimes used for ear infections, topical skin infections, respiratory infections, and certain examples of these are erythromycin, clarithromycin, and azithromycin. Those are some of the most common ones that you'll see out there. And again, of course, there are always contraindications, meaning times that they shouldn't be given. But for the most part, the macrolides are a good choice when we're talking about a person, or really anyone, who might be sensitive to other types of treatments or who wouldn't be able to withstand one of the harsher antibiotics. Our last group of antibiotics are the folic acid synthesis inhibitors. Now before, I called these metabolic pathway inhibitors, and they are that. They do inhibit metabolic pathways. But on this slide, I wanted to be a little bit more specific and mention which metabolic pathway they work to inhibit. These types of drugs are oftentimes called the sulfa drugs. One example is sulfonamide. And they are manufactured entirely in the lab. These are completely synthetic antibiotics, and they're based off of compounds that are found in dyes. What these type of antibiotics do is stop certain metabolic pathways from functioning correctly. More specifically, what they do is they block folic acid synthesis in bacteria. Folic acid is an organic compound that acts as a coenzyme for many other enzymes. So by stopping the synthesis of folic acid, what we're doing is creating a cascade effect in the bacterial cytoplasm and interfering with all kinds of other metabolic activities as well. So what this will do is, if this is the pathway that is used to create folic acid, this type of antibiotic will come in and interfere with one of those steps, whether it is at the first step or the second step. In the end, either way, what ends up happening is folic acid is not created. And what that means is the bacteria is then not able to carry out a variety of other metabolic activities because its enzymes won't work. If folic acid is not made and folic acid works as a coenzyme to these other enzymes for other metabolic activities, then those metabolic activities will cease because their enzymes won't function properly. These are typically fairly broad spectrum medications. They work on lots of different types of bacteria because all bacteria need folic acid to carry out many of their activities. And most of the time they are bactericidal. Because they do interfere with a lot of metabolic pathways, and as a result, the bacteria stops being able to carry out the activities they need to stay alive. So most of the time, these types of drugs help kill the bacteria fairly quickly. The downside to these types of medications, or at least one of the downsides to these types of medications, is that a lot of people are allergic to them, and so they are not used as frequently as other types of medications a lot of times because of the risk of allergies. So far in this lecture, we have talked about what antibiotics are and how they work against bacteria, what exactly it is that they do to interfere with the growth of bacteria, or how it is they just kill them outright. And now we're going to take a step in another direction and talk a little bit about a phenomenon called antibiotic resistance. And when I say this term, it's important to understand that there are sometimes a lot of misconceptions around it. And so just to start things off, I want to make sure everyone is on the same page and working with the same definition of what antibiotic resistance is. Antibiotic resistance is a phenomenon that occurs when an antibiotic that should work to kill a certain type of bacteria does not work anymore. So what that means is that if we have a situation where antibiotic A should be able to work against bacteria B, but it doesn't, for whatever reason, that antibiotic is not able to kill that bacteria, then we have a situation that we call antibiotic resistance. It's important to understand that this really has nothing to do with the person. A lot of times people think it is the person becoming immune to the drug, but that is not what's happening in this situation. In this situation, we are talking about a specific phenomenon that occurs when an antibiotic does not work to kill a certain type of bacteria, even though it should work. And this phenomenon of antibiotic resistance is a very serious issue facing our society and societies all over the world. This isn't just something unique to the United States. This is occurring worldwide. And although it does occur worldwide, we're just going to look at some United States statistics right now. In the United States, out of all of the bacterial infections that occur during any given year, about 2 million of them are caused by bacteria that are resistant to one or more antibiotics. This causes sometimes long-term health complications for people, and occasionally there are situations where no antibiotics will work to help someone and they end up dying from their bacterial infection. So again, in the US, about 23,000 deaths per year are caused by bacteria that are resistant to antibiotics. And then if we kind of zoom back out and look at all of this worldwide over the next about 35 years, it is expected that there will be about 300 million deaths and illnesses as a result of antibiotic resistance. So basically there will be about 300 million people who get sick and or die over the next 35 years as a result of bacterial infections that cannot be treated with antibiotics anymore because the bacteria are resistant to the antibiotics. This poses a pretty serious issue for scientists and healthcare providers to figure out and to deal with. Antibiotic resistance affects everything from things like military readiness and food safety and security to economic effects. It's expected that there, again, over the next 35 years or so, there will be about $100 trillion in losses, again, worldwide, due to the phenomenon of antibiotic resistance. Basically, what that figure includes is everything from people missing days of work to deaths to all of the impacts that these types of illnesses and deaths will have on the worldwide economy. So this is a very, very serious issue. And hopefully these predictions will not come true. Basically, these are kind of based on if we go along the path we've currently been on, this is what we can expect. However, hopefully there will be a lot of research in the next decade or so about antibiotic resistance, and there will be some discoveries and some breakthroughs related to this phenomenon. On our last slide, we talked a little bit about what antibiotic resistance is and what the effects of that are and may be if we don't get it under control. And on this slide, I want to talk a little bit about the mechanisms and go into the actual details of what makes it so that a bacterium can resist antibiotics. So again, I've got a definition up here. This is the same basic definition as the one I gave you on the previous slide. But basically, antibiotic resistance is the ability of a bacterium to not be killed by one or more antibiotics. And again, this doesn't refer to a person becoming immune to the drug. In this situation, the person has nothing to do with it, really. It's all about the fact that the bacteria is not susceptible to the drug. So when we are talking about this phenomenon, we oftentimes say that a bacteria is susceptible or sensitive to an antibiotic. If that bacteria is killed by that antibiotic, or at least its growth is stopped. And if a bacterium is resistant to that antibiotic, it means that antibiotic does not work to kill that bacteria. So make sure you are clear on those terms before you go any farther in the lecture because they are really important. And just to reiterate one more time, if a bacterium is sensitive or susceptible to a certain antibiotic, it means that antibiotic works to either kill it or stop its growth. And if that bacteria is resistant to that antibiotic, it means that that antibiotic does not work to kill the bacteria or stop its growth. So basically, if you have a bacterium, we'll call it bacterium A, it might be susceptible to antibiotics 1, 2, and 3, but resistant to antibiotics 4, 5, and 6. So if we have a patient who is infected with bacterium A, we know we could use antibiotics 1, 2, or 3, but not 4, 5, or 6 because those won't work. The opposite could be true for bacteria B. Bacteria B could be susceptible to antibiotics 4, 5, and 6 and resistant to antibiotics 1, 2, and 3. So if we have a patient who is infected with bacterium B, we want to use antibiotics 4, 5, or 6, and not antibiotics 1, 2, or 3. So all of that so far that we've said goes with this concept of the fact that the characteristic of resistance goes with the bacteria, not with the person or not with the antibiotic. Resistance is a characteristic of a bacterium, not a person, not the antibiotic itself. So hopefully that is clear. If it is not clear, stop right now and send me an email or do something along those lines so I can work with you on that topic before you continue. For those of you that it is clear to, let's talk about what the mechanisms of antibiotic resistance are. And what that means when I say the mechanisms of antibiotic resistance, what I'm talking about is some kind of action that a bacteria takes to make it so that an antibiotic does not work to kill it. So when we talk about the mechanisms of antibiotic resistance, we're talking about the activities that bacteria do, or sometimes can do, in a certain situation to make it so an antibiotic does not work to kill it. There are 5 main ways that bacteria can make it so that an antibiotic will not kill it. The first way is to develop a new enzyme or some kind of alternative to an existing enzyme. These enzymes may then work to break down the drug. In the center here, you see a resistance plasmid, and we've talked about plasmids before and how they can contain genes for antibiotic resistance, and now we're getting into some of the details of that. So this is our bacteria here, and it has a plasmid that has traits on it for antibiotic resistance. One of the traits, or one of the genes that this plasmid has, codes for an enzyme that will break down antibiotics. So if this bacterium starts out its life and doesn't have this plasmid, antibiotics are going to work to kill it. But if it acquires this plasmid through something like conjugation or transformation or some other kind of event, then it all of a sudden has genes that allow it to resist antibiotics. And these genes that are shown here, one of them allows this bacterium to make an enzyme that will degrade the antibiotic. So the antibiotic comes into the cell and the bacterium creates this enzyme to break apart that antibiotic so the antibiotic cannot do its job anymore. Now, another way that a bacterium could be resistant to an antibiotic, or another mechanism it can use, is to prevent the drug from entering the cell. So it may inhibit the uptake of the drug. Now that picture is not exactly shown on this diagram, but I can just kind of draw you a picture of it. This is a bacterium, and we have, we'll say, antibiotic D, because the other letters are taken already. And I'm just going to make it a little star, because those are easy to draw. And normally, antibiotic D would bind to something on the outside of the bacterial cell and be taken inside. But what this bacterium will do is just kind of alter something on the outside of its cell, so there's no place for that antibiotic to bind. And as a result, if the antibiotic doesn't bind to the outside of the cell, it can never make it inside the cytoplasm and can never do its job. The third mechanism of antibiotic resistance is the development of drug transport pumps, and that is one that is shown here. We have our resistance plasmid in the center here, and one of the genes on that plasmid codes for this antibiotic degrading enzyme. Another one, though, codes for a protein that acts as what we call an efflux pump. So in this situation, antibiotic C comes into the cytoplasm, makes it in, no problem, but it doesn't stay there very long because the bacteria has created this tube-like protein that it inserts into its cell membrane and cell wall that ends up pumping the drug out. So the antibiotic comes in, but doesn't stay long enough to be able to do its job. It just gets pumped right back out by the bacteria. Another way that an antibiotic or that a bacteria might become resistant to antibiotics is that it alters its binding sites. So antibiotics, especially the ones that bind to ribosomes, have a very specific location that they are looking to bind. And what a bacterium might do is either change its own ribosome just slightly so that the antibiotic cannot bind, or it might create an enzyme that actually just alters the antibiotic itself just a little bit. So then the antibiotic doesn't fit on the ribosome or doesn't interfere with peptidoglycan anymore, or doesn't interfere with the enzymes involved in DNA replication. Basically, there are a lot of different things that the bacteria can do to alter either the antibiotic or the location where the antibiotic is supposed to bind so that that attachment cannot be made. And if that attachment cannot be made, then the antibiotic doesn't work. And then the last one that they can do, and this is actually fairly easy for them, is just to alter their metabolic strategy or pathway slightly. This is especially common when we're talking about resistance to things like sulfa drugs, which we talked about earlier, which are the metabolic pathway inhibitors. And oftentimes, bacteria have multiple ways of arriving at the same result. So they may prefer to use one pathway to make folic acid, or one pathway to make ATP, or one pathway to make enzyme X, whatever it is. However, they oftentimes have the ability to get to the same result using a different pathway or different set of pathways. So one really good example of this is for humans, we basically have one way to make ATP on a long-term basis, and that is making sure we have access to oxygen so we can go through aerobic respiration. Now, many bacteria also go through aerobic respiration, right? They use that pathway to make ATP as well. But many bacteria also have the ability to use fermentation very well, or use anaerobic respiration very well, or use aerotolerant pathways. So they don't necessarily have to have oxygen to make ATP. Some of them do. The obligate aerobes obviously do. But many of them have the ability to get to their end result, which in this example is ATP, in multiple ways. And so for bacteria, if you give them an antibiotic that interferes with one metabolic pathway, they may simply be able to kind of circumvent that and go around that metabolic pathway to still get to whatever is required. So in the example with sulfa drugs, remember we were talking about how if you give them a sulfa drug, that interferes with folic acid synthesis. And that might work in many cases, but for other bacteria, they may be able to have an alternate pathway to make folic acid. So you give them an antibiotic and that inhibits one of their pathways to make folic acid, but it doesn't inhibit another pathway that they might have that allows them to arrive at the same result, which is creating folic acid. So for bacteria, a lot of times it is relatively easy to kind of circumnavigate or circumvent these antibiotics that normally would prevent a metabolic pathway from happening. They'll just use a different metabolic pathway and get to the same place that they wanted to go before. So now we have talked about what antibiotic resistance is. And remember, antibiotic resistance has to do with the bacteria, not the person, not the antibiotic, exactly. And we've talked about what the things that bacteria can do are to make it so that antibiotics don't work, right? So antibiotic resistance is the ability of a bacteria to not be killed by an antibiotic. And there are multiple things that they can do to make it so that they are not killed, whether that involves using an alternate metabolic pathway, creating a new enzyme that degrades the antibiotic, pumping it out of the cell, just altering the binding site of the antibiotic or of themselves slightly, etc. And now we want to talk about how it is that antibiotic resistance develops in populations or colonies. On our last slide, we talked about the individual activities that an individual bacterium can carry out to make it so an antibiotic doesn't work to kill it. But on this slide, we're going to be looking at the bigger picture and understanding how we get huge colonies of resistant bacteria that will end up causing problems for people. So the first thing to understand is that antibiotic resistance exists naturally in many populations of bacteria. Oftentimes, bacteria just naturally have genetic sequences or ones that have been changed during recombination events like conjugation, transformation, through transposons, etc., that allow them to resist antibiotics. The antibiotics themselves do not cause the bacteria to become resistant. What they do is when you introduce an antibiotic into a population of bacteria, that acts as what we call a selective mechanism or a selective factor. That antibiotic creates a situation where the bacteria that are naturally resistant to the antibiotic will survive and reproduce, and the ones that are naturally susceptible to the antibiotic will die off and not reproduce. So overuse of antibiotics ends up selecting for resistant individuals. What that means, again, is that in any given population, and I'm going to use this one that I've created here as an example, most of the bacteria will be susceptible to an antibiotic. We'll call the yellow ones the susceptible ones. But you might have one or two that are naturally resistant to the antibiotic. When you put the antibiotic into this bacterial culture, the susceptible ones, the yellow ones, are going to die off, but the naturally resistant ones are going to live and reproduce. Eventually, this leads to entire colonies that are resistant to the antibiotics. Then what ends up happening is the resistant ones spread like they normally do through people's bodies or from person to person, or in some cases from animals to people. And what we end up having is a person or sometimes an animal that is infected with an entire population of bacteria that is resistant to the antibiotic. So I have created an animation for you to help you understand this. I'm going to play it now, and we can go back over it again through email or through office hours if you need to work on that. So starting off, again, like I said, we have our population of bacteria, right? This petri dish is our population of bacteria, and it is made up of individual organisms. The yellow ones are susceptible to the antibiotic, meaning that when the antibiotic is introduced, that antibiotic will kill them. The blue ones, or the teal ones, are resistant, meaning that even though the antibiotic has been introduced, they are going to survive. So we start out with a mixed population. Basically, we've got most of them are susceptible and just one or two that are resistant. We introduce the antibiotic, and that antibiotic is going to work to kill off the yellow ones, but not the blue ones. So the yellow ones are dying here, but the blue one's just fine. It's naturally resistant, either because it can pump out the drug or whatever. The yellow ones then are gone, and the blue one is left. The blue one then will replicate. It will go through binary fission because there's nothing interfering with that, and it will create more just like itself that are naturally resistant to the antibiotic. So if we introduce the antibiotic again, nothing's going to happen. That antibiotic is not going to work to kill them because they are all just like the first one, They're resistant to the antibiotic. Now what we have is a growing population of individuals that are all resistant to the antibiotic. No matter how much of the antibiotic we give them, it doesn't work to kill them. And now we have an entire population that is resistant to the antibiotic instead of just one. Now, this population, if it were to get into a person's body, would still be resistant to that antibiotic. And if we tried to treat that person with the antibiotic, it would do nothing to help them deal with that infection. Now, so far, all of our lecture really has focused on the antibiotics. And the reason for that is because out of all of the antimicrobials prescribed, at least in the United States, and honestly probably worldwide, antibiotics are most of them. So most of the time when people get infections, they are either bacteria or viruses, and we don't have a lot of antiviral medications. We are going to talk about how they work later on this slide, but for the most part, when people get infections, especially in the United States, they are either bacterial infections or they are viruses. We have things to treat bacterial infections called antibiotics, and in some cases we have antiviral medications that will treat viral infections, but most viral infections we just kind of have to let run their course. So most of the antimicrobials that are prescribed are antibiotics, and that is why I spend so much time talking about those. However, there are other types of microbial infections. There are fungal infections, there are protozoal infections, there are helminthic infections, and there are viral infections. And on this slide, we're going to talk a little bit about how each of those types of medications tend to work. Because chances are good at some point in your healthcare career, you will run into a situation where you do have a patient who has or has had a fungal infection, maybe even a protozoal infection, a helminthic infection, and certainly a viral infection. Antifungals are the medications that we use to deal with fungal infections. Fungal infections are relatively difficult to treat, especially when we compare them to bacteria, because bacteria are prokaryotes and humans are eukaryotes. On the other hand, fungi are also eukaryotes. So that means we have to create a drug that will work to damage one type of eukaryotic cell, the fungus, and not do too much damage to another type of eukaryotic cell, which is the type of cell found in humans. So fungi are a little bit harder to treat than bacteria because fundamentally our cells are very similar. So fundamentally, fungal cells and human cells have a lot of similarities, and so it's a little bit more challenging to create medications for them than it is for bacteria because they are very different cell types than the types found in humans. Over the years, though, we have found enough differences between fungal cells and human cells that we have been able to create some very effective medications. And the medications that we use to treat fungal infections work in three main ways. Most of them work to interfere with fungal membranes. Remember, in eukaryotes, there is a special type of lipid that is found in the cell membrane, that's not found in prokaryotes for the most part, called a sterol. Sterols are lipid molecules, they're ringed lipids that help provide stability and flexibility to the eukaryotic cell membrane. In humans, the sterol that is found in our membrane is called cholesterol. But in fungi, the sterol that is found in their membrane is called ergosterol or ergosterol, depending on who you're talking to. Either way is fine. So many of our antifungals work to interfere with ergosterol, this sterol that is found in fungal membranes but not in human membranes, and destabilize that membrane. So our cells end up being fine because we have cholesterol, and that is a different type of sterol molecule. but our antifungal membranes will interfere with the membrane of the fungus by focusing on ergosterol. Other things we can do in terms of antifungal activity is to inhibit chitin synthesis. Remember, chitin is the molecule that makes up the cell wall in fungi. And humans don't have cell walls in their cells and don't make chitin, so that type of drug really doesn't interfere with our cells at all. But we can interfere with the cell wall in fungi, and that will at least help stop their growth so that our immune system can do the rest of the work. We've also found that there are enough differences in the ways that proteins are made in fungi and humans that we can target their protein synthesis in a similar way to the ways antibiotics work when we're talking about the protein synthesis inhibitors. So there's enough difference between the ribosome in a fungus and the ribosome in a human cell that we can target their protein synthesis by using certain medications and not interfere with ours too much. Now, our next type of drug here is the antiprotozoal. Antiprotozoals are medications used against protozoal infections, and protozoa are actually even harder than fungi to get rid of because when you compare a human cell to a protozoal cell, they are very, very similar. So they're even more similar than human and fungi cells. And again, that makes it very difficult for us to create drugs that will target the protozoa or target that microbe and not harm our cells as well. However, our antiprotozoals often work in ways that we take advantage of the fact that most of the time protozoal cells are much smaller than human cells. These type of drugs end up becoming activated at very high concentrations. So this one I couldn't find a great picture of, so I'm just going to go ahead and draw it. If this is our human cell and our protozoa is inside, and you put in a medication, so our medication is coming inside, and you put in 100 particles of medication, right? 100 of them go in there. I'm not going to be able to draw 100, but that's just a good round number. Then we maybe have 10 or so that end up staying in the human cell cytoplasm. 90 of them end up getting into the cytoplasm of the protozoa. And so even though the human cell also has this drug inside of it, it's at a very low concentration, relatively speaking. But if we look at our protozoa and we see all of the individual particles of medication that are in there, we can see that that's a much higher concentration. That very high concentration of the medication starts to interfere with metabolic activities of the protozoa, and it may not be able to do things like make ATP, break down glucose, or do any number of metabolic activities it needs to because that medication is basically just getting in the way of it. Now, our next type of medication are the antihelminthics. These are very difficult for us to develop. They are one of the hardest types of drugs for us to develop because helminths are fundamentally animals, and humans are also animals. And our cells have very little in terms of differences between them. They are very, very similar. And so it is quite challenging to develop antihelminthic drugs because the cell structure of a helminth is almost identical to the cell structure of a human. Obviously, we're very different organisms, but at a cellular level, at a fundamental cellular level, there aren't a lot of difference between the cells we find in the human body and the types of cells that we find in helminths. But there are enough small differences in the microtubules that we will find in helminths and the enzymes that they use to break down glucose so that they can go through glycolysis, that most of our drugs work in one of these two ways. So most antihelminthics will work either to inhibit microtubule function in worms, and oftentimes that kind of paralyzes them or stops them from being able to go through cell division, so that even if it doesn't directly kill the worm, it prevents it from replicating, or it kind of paralyzes it so our body can do the work of getting rid of it then. Or the medication will work to interfere with their ability to use glucose to make ATP. And so that ends up killing the worm pretty quickly because they also need to make ATP just like we do. And if we interfere with their ability to make ATP, then they will die fairly quickly, and our body will be able to do the work to expel the worm after that. Now, our last type of medication is the antiviral. And this is, for the most part, the most difficult type of drug to develop. The reason for that is, remember, viruses are what we call obligate intracellular parasites, meaning they must be inside a living, metabolizing, active host cell to be able to do anything. And so what that means is we have to, for the most part, develop a drug that we can get inside a human cell that will damage just the virus and not any of our internal cellular components. And that is quite a challenge. That is one of the reasons we don't have a lot of antiviral medications. The antiviral medications that we do have, though, work to stop the virus from replicating. So they work in some kind of way to either inhibit the binding of the virus to the receptor on the host cell. So maybe they interfere with adsorption. They might interfere with the capsid coming apart. So the drug will interfere with the capsid coming apart, and therefore the viral genetic material can't get into the cytoplasm of the cell and can't replicate. They might interfere with synthesis of the viral nucleic acid. They might interfere with the capsid proteins themselves when they're getting put together. They might interfere with the ability of the virus to be able to leave the cell. So each of our antiviral medications works a little bit differently, but what they all have in common is that all antiviral medications work to stop one of the stages of viral replication. Whether that is the beginning stage and they block adsorption, or one of the middle stages where they're blocking synthesis or assembly, or the end stage where they're blocking release of the virus. In some way or another, all antiviral medications work to stop the virus from replicating at one of the stages.